Hello, everyone, and thank you for attending today's webinar on Agilent BioTek Lionheart technology, driving physiological relevance in early drug discovery, presented by Dr. Charles William Amir Mansour, Global Business Development Manager here at Agilent BioTek. Before we begin, I want to cover a few housekeeping items. At the bottom of your screen, there are multiple application engagement tools that you can use. All of the engagement tools are resizable and movable, so feel free to move them around to get the most out of your desktop space. Today's presentation will appear in the Media Player module, followed by a live Q&A session moderated by Dr. Margaret Pruitt, a product manager for Agilent in the Cell Analysis Division. And if you have any questions during the webcast, you can submit them through the Q&A engagement tool. We will do our best to get to as many of your questions as possible at the conclusion of the presentation. The rest will be answered later via email. And please know that we do capture all questions. Additional help materials are available in the resource list. We encourage you to download any resources or links that you may find useful. For the best viewing experience, we recommend using a wired internet connection and closing any programs or browser sessions running in the background that could cause issues. Webinars are bandwidth intensive, so closing any unnecessary browser tabs can help conserve your bandwidth. For the best audio quality, please make sure that your computer speakers or headset are turned up and the volume is on so you can hear the presenter. Please note that an on-demand version of the webcast will be sent to you by email in the following days. Again, I thank you all for joining us today, and with that, I will turn things over to Charles.
Shouldn't your instrumentation support the breadth of your research and see you through to publication? Hi, I'm Charles William Amir Mansour, Global Business Development Manager with Agilent BioTek. Today, I'd like to provide some insight and thought-provoking content, which will hopefully get you thinking out of the box with new ideas to take your research to the next level. So, over the past decade, the way we think about the drug discovery process has evolved with the development of a wide range of complementary assay workflows to achieve therapeutic strategies. One idea addresses the physiological relevance of cell models used in early drug discovery. The belief here is that models that more closely resemble human physiology will provide higher efficacy during clinical trials. Changes in phenotype associated with this biology is obviously best evaluated by quantitative microscopy. A second approach relies on the use of biochemical-based assays to study the mechanism of action of these test molecules or drug therapeutic candidates. Now, these two strategies are complementary to one another and rely on the use of multimode reader detection optics and quantitative microscopy optical systems. And with that in mind, BioTek developed the citation platform quite a number of years ago. And this caters to the needs of research groups that want to take this diversified and multidisciplinary approach using multimode reader optics, wide field microscopy, and filter-based optics to drive experiments in parallel to run mechanistic studies and rich phenotypic assays. And the latest addition to the citation platform or suite of platforms is the Citation C10, which also offers spinning disk confocal fluorescence optical capabilities, which now allows you to look to much better 3D temporal resolution imaging applications and look at 3D spheroids, tumoroid, organite structures, or biology thick tissue biology applications. So a multidisciplinary approach by using these technologies. Having said that, and to cater to the needs of researchers focused on imaging-based workflows alone, we developed the Lionheart FX and LX platforms. The Lionheart LX is designed to be an affordable solution to drive automated wide field microscopy workflows. And the FX system is optimized for live cell imaging applications when configured with features like an environmental control cover, temperature control, CO2, O2 gas controller, humidity chamber, and an optional dual reagent injector. Now, both of these platforms are equipped with a 6-position objective turret and a 16-bit CMOS wide field of view camera. And what one inherits by having this optical design is being able to tap into a range of modalities, imaging wide field modalities, and clearly a broad spectrum of workflows. So here we can see the range of modalities that are applicable to the Lionheart platform: fluorescence, wide field imaging, bright field, high contrast bright field, phase contrast, and color bright field. And towards the end of my presentation, I will go through a number of workflows which tap into each of these modalities and how they're utilized to look at various phenotypic and morphological changes that occur in our biological systems that are being evaluated. Now, one of the many benefits and attributes of using the Lionheart system is that you can utilize a broad array of filter cubes that are designed to cater to imaging fluors across a broad spectrum of light. So from the blue end of the spectrum, say DAPI labeling for cell nuclei, all the way out to the red end of the spectrum with Psi7. So these are LED excitation filter cubes, which have excitation emission filters and dichroic mirrors that are optimized for different fluorophores. And that means that having access to a huge library of filter cubes gives you, the user, the flexibility to label your biology based upon the target of interest. Now, the other aspect of application diversity, which is very important, is being able to visualize that biology. And with the Lionheart platform, you have the ability to utilize objectives from 1.25x all the way out to 100x oil immersion objectives. And that then means that we can look at whole organisms, endpoint assays, subcellular detail, and so much more. So the application flexibility is further expanded by having the ability to utilize objectives of this nature, and both air and oil immersion objectives, spanning a broad magnification range. Now, as many of you know, the refractive index is a measure of how much of a material, or let's say media, will bend light as it travels through it. Microscope objectives are engineered to have a refractive index of 1.52. Air objectives and samples that reside in, let's say, water or PBS introduce a mismatch in refractive index that causes a refraction in the light path, resulting in image distortion. Think of a straw in a glass of water. I think that's the best analogy I can think of. Now, immersion oil objectives attempt to match the refractive index between the objective and cover slip interface. And for that reason, we've developed oil immersion objectives, or we use oil immersion objectives that will be important to do high resolution imaging in the 60 and 100x range. And what we're looking at here is high resolution 100x oil immersion images of primary mouse neurons cultured from embryonic mouse cortex. From a staining perspective, in the blue channel, we've got a DAPI stain, of course. Green channel is MAP2 antibody stained, localized to neuron dendrites. Magenta pink channel is GFAP antibody stained localized to glia cells, or obviously non-neuronal. And in the red channel, beta-3 tubulin stained localized to neuronal axon and dendrite compartments. And portions of the image on the right shows red staining alone. Therefore, this indicates neuronal axons. So really impressive high resolution images using oil immersion 100x objectives. Now, one of the other attributes of the design, I would say, of the Lionheart is having an open stage architecture, which has many benefits to it. One of it is being able to couple or integrate it with a perfusion-based system. So the millipore Onyx system allows you to drive a number of workflows, which allows you to perfuse cells, treat cells with various test molecules or agents, and look at changes that occur in that cellular model. You can even homebrew, or I would say with this architecture, it's compatible with a homemade system. So you can feed tubing through the stage via an access port, and that allows you to obviously expand the footprint of applications that would otherwise be very challenging or difficult to do. Now, live cell applications are becoming so much more relevant nowadays than they were many, many years ago, and so that was important in the design of the Lionheart system. So features such as temperature modulation or control, gas control, both CO2 for live cell assays, O2 for, let's say, hypoxia studies, and then having the ability to utilize a dual reagent dispenser to add rate limiting reagents to instigate reaction. These are great features to have that further expands the footprint of applications that would otherwise be very challenging, if not impossible. So calcium mobilization, ion channel studies, hypoxia oxidative stress oxidative burst studies, and so having these capabilities, I think, can be of huge benefit to exploit a wider spectrum of applications. So here we're looking at examples of applications and workflows that many of our customers utilize the Lionheart system for, from 3D cell biology workflows all the way out to neurobiology-based workflows. So an incredibly diverse spectrum of assays become possible with this type of optical architecture and features, hardware features that are all possible with the Lionheart system. Now, when you think about building a comprehensive scientific story, a single experiment never exists in a vacuum. It's always part of a larger hypothesis. To publish a comprehensive story, you need to show compelling screening data backed up by individual dose response experiments to justify your choice of target. Now, live cell assays from 2D and 3D cell culture models can clearly illustrate the phenotypic and morphological changes that occur. And microscopy allows you to visualize proposed mechanisms at work. Now, further mechanistic studies can be explored using fixed cell marker staining and quantification. And finally, the physiological relevance of the proposed interactions need to be confirmed by histological evaluation of animal models or patient samples. And so with the Lionheart platform, you're able to build a comprehensive story from drug screening through to histological confirmation by taking a multidisciplinary approach with complementary imaging workflows and technologies. Now here what we're looking at is really the spectrum of capabilities offered by Gen 5, which is the software platform that drives the Lionheart system. And we have a huge array of parameters, tools, functions, and algorithms, high content algorithms built into it that allows us to go from image capture through processing, analysis, and being able to even set up customized analysis metrics based on individual needs of the user, all the way out to publication. Full array of capabilities that takes you from A to Z with Gen 5. Now, over the past several years, we've worked diligently with many researchers across a number of industries and institutes to drive a broad array of applications and workflows. And here are, I would say, here's a sampling of the most routine applications that we see quite often where the Lionheart is being utilized. Now, we won't have time to cover off every single topic shown here, but I've cherry-picked a number of topics where we see quite often, and I wanted to provide a little bit of insight and some thought-provoking content that'll get you thinking about experiments that you may want to design and explore taking this multidisciplinary approach. In this example, we're looking at a 3D cell culture assay, specifically tumor invasion, using a surrogate model for metastasis by co-culturing either U87 or LN229 glioblastoma cells with primary fibroblasts. The workflow entails the use of 96-well round-bottom microplates to promote spheroid formation. After we co-culture the two respective cell types and allow them to aggregate, we gently remove the media and replace it with cold matrigel containing various concentrations of inhibitor. And we allow that matrigel to gel around the spheroid. Media then was added on top of that matrigel layer containing various inhibitors in question. And then the spheroids were imaged every 12 hours in the bright field channel as a montage Z-stack to cover the spatial depth of that entire spheroid structure. Then we carry out cellular analysis using the architecture of Gen 5 software by placing an accurate and detailed primary and secondary object mask around the entire 3D spheroid and invader podia structure. Now, when we plot that kinetically over time, as you can see in the bottom right-hand corner of this graph, we can clearly see the difference in the invasive characteristics of the two glioblastoma cell types, where U87s are highly invasive in metastatics, and LN229 on the other hand, exhibit much lower metastatic characteristics. So another great example of a 3D cell culture assay. Now here we're looking at two cellular processes, apoptosis and necrosis, where we're utilizing a Lonza raft cell culture model. For those of you not familiar with the raft system, raft stands for real architecture for 3D tissues. And it's essentially a collagen hydrogel based 3D cell culture model where cells are embedded into a consistent 120 micron thick gel layer. The methodology itself involves the addition of human colon carcinoma HCT116 cells to the raft hydrogel and allow to propagate to form 3D tumoroids. Now, following the creation of those tumoroids, natural killer cells are introduced, which have been previously stained with a deep red cell tracking dye. And, you know, this was added to the media above the hydrogel layer. At the same time, components from Abcam's kinetic apoptosis kit is added to each and every well. And that kit contains a polarity sensitive indicator for viability and apoptosis, which fluoresces green, so it's a green indicator, and propidium iodide, which is your necrosis stain, and that fluoresces in the red channel. Now, at that point, we carry out kinetic imaging Z-stack imaging, I would say, over the course of 5 days and trying to observe or detect the effects of the natural killer cells on the HCT116 tumoroid population. Now, the graph shown in the bottom right corner basically shows us two different cell culture models, two in a 3D cell culture model, and trying to observe the effects that these natural killer cells are having on our HCT116 population. And we can clearly see the differences between those two cellular systems, where in a 2D model, the cells become lose viability and have become necrotic over time and die. And it really proves or demonstrates, I would say, why a 3D cell culture model provides much better target cell viability data over a long-term incubation, in this case 5 days, and therefore it's a much more robust method for looking at viability. Now, another application which is extremely routine in the course of using a Lionheart system is counting objects or counting cells. Now, in the context of this workflow, which is a plaque assay, we're looking at quantifying GFP expressing viral plaques in a cell monolayer. In the context of this application, we've got a 96-well plate, and we're taking a montage of images covering the entire area of every single well, stitching those images together, and then automating the process of placing an object mask around each individual GFP positive viral plaque and counting the population. So whether it's fluorescently expressing cells or labeled cells, very easily done in terms of acquisition, processing, and analysis to be able to count populations of objects or cells. Now, having said that, there are many workflows where we're monitoring proliferation of cells over the course of several days or possibly weeks, but the cells are not expressing a fluorescence reporter, nor do we label the cells. So these are unlabeled cells in their native state or unperturbed. And in this example, we're kinetically monitoring cell counts every 2 hours using high contrast bright field. So with the high contrast bright field modality of the Lionheart system, the light is transmitted through the well, and when it hits each and every single cellular object, it's refracted. That light is refracted, creating a bright spot. So you're going to have hundreds or thousands of bright spots. And it makes it so easy because you have this nice contrast between your background and foreground being the cells, to then automate the process of placing an object mask around every single bright spot and count the total population of cells. So whether we're dealing with labeled biology, fluorescently expressing cells, or unlabeled cells, we can very easily automate the process of image capture, processing, and analysis using the hardware and software architecture. Now, other applications which are very common across our user base, I would say would be looking at cell migration. And, you know, the application that we're looking at here is very much applicable to the field of angiogenesis, wound healing, inflammation, oncology. And so if you're looking at the migration of cells, we certainly have the ability to tap into those workflows. So in this particular application, we're using an Oris Pro plate sourced from a company called Platypus Technologies to investigate cell migration using a GFP expressing breast cancer cell line. And so the plate itself has a biocompatible, biodegradable gel printed in the XY center of each well. The cells are seeded into each well. The cells will settle and attach to the bottom of the plate. Once the media is in contact with that biocompatible gel, it breaks down and reveals a cell-free zone, an area where there are no cells at the beginning of that time course. Now, we can then treat each well with compounds or test molecules that either facilitate or inhibit cell migration. And then the Lionheart system is then used to determine, or I would say Gen 5 is used to determine the percent of confluence of cells migrating into that cell-free zone using an algorithm within the architecture of the software. This obviously allows us then to assess the ability for a drug or test molecule to induce or inhibit cell migration and calculate the efficacy of that test molecule. So cell migration is clearly another application that's very easily driven using the architecture of our system. And that's whether it's cells that are expressing a fluorescence reporter or labeled, or whether it's unlabeled cells. We certainly have the ability to carry out cell migration, wound healing, or scratch assays using the Lionheart system. Now, there are also applications where the changes that are occurring are not over the course of days, or I would say hours, days, or weeks, but milliseconds or seconds. So an example of that would be calcium flux. And in this example, we're looking at how we're using the onboard dual reagent injectors to treat HeLa cells with ATP to elicit a calcium flux, and the changes in fluorescence are then monitored by rapid fluorescence imaging. We then use those images, kinetic images over time, to create timestamp movies and look at those kinetic images from the same well location over that time course. And we're obviously using parameters and algorithms in Gen 5 software to carry out cellular analysis to identify cells that are positive for calcium flux and provide kinetic curves from different experimental conditions. So another example of a kinetic-based workflow. Now, other applications I've seen quite often where many of our customers utilize Lionheart technology for would be the area of looking at DNA damage or DNA repair, so a genotoxicity-based workflow. And genotoxicity applications are generally used to study the mechanisms of DNA damage and repair. So what we're looking at here is a COMET assay, or another term that it's referred to as is a single cell gel electrophoresis workflow, which is a direct measure of DNA damage. So what happens in this workflow is cells are exposed to a test compound, test molecule, which are embedded in an agarose gel, lyes to remove membranes and histones, treated with an alkaline solution to unwind and denature the DNA, and then subject it to an electrical current or field to separate fragmented DNA from intact DNA based on the charge to mass ratio. And then following that electrophoresis step, comets are stained with a DNA dye like cyber gold or green, in this case cyber green, and then we basically image those comet slides in the GFP channel using a 2.5 or a 4x objective. And then image analysis is performed where the GFP signal emanating from the comet head is used to place a primary object mask around that head. So that's the intact DNA. And then a secondary object mask is then established to extend from the primary mask to capture the comet tail. That's the fragmented DNA. And then total GFP signal and length measurements for the primary and secondary masks are used to calculate very common metrics used in comet assays, like what's the percentage of DNA in the comet tail and tail moment. So a nice example of a genotoxicity comet-based assay, I would consider it high throughput to look at DNA damage or repair. Another application that many of our users have been exploring is using a RNA FISH, fluorescence in situ hybridization workflow, to look at gene expression. And in this example, we're looking at an Affymetrix view RNA in situ hybridization assay method, which starts by fixing and then permeabilizing and digesting the cells with a protease to access the mRNA target. And then a set of target-specific oligoprobes are used to hybridize to each mRNA. And an oligo signal amplification system is then used, which hybridizes to the specific mRNA probe based on its sequence specificity. And then last but not least, the final step being the addition of an oligo conjugated fluorescent probe mixed with, it's a mixture containing various Alexa fluors, so Alexa 488, 546, 647, 750. And, by taking this multi-labeled approach, basically fluorescent signals that you obtain or capture are specific to the starting mRNA probe. Now, we also DAPI nuclear stain, that's required so that we can count the total populations of cells. Now, fluorescence images are obviously captured with the Lionheart system, processed to remove the background signal. And then we use tools like a scatter plot analysis tool in Gen 5 software to determine average standard deviations of the signal compared to negative control wells. And then these values are used to define the subpopulation threshold values in the different fluorescent channels that are obtained to identify positive responding cells per image. Now, when we plot, so when the resulting data is plotted against the concentration of treatment added to the wells, the final curve that you obtain can be used to determine the effects of these test molecules to either stimulate or inhibit expression at the RNA level with single cell resolution, and allowing you to look at the heterogeneity of that response. So again, another wonderful example of a completely different application looking at gene expression using fluorescence in situ hybridization methodology. In this example, we're looking at an immunofluorescence application where we're demonstrating the ability of the Lionheart system to capture images through various stages of the cell cycle using these really large mammalian cells derived from male rat kangaroo epithelial kidney cells. They're called PTK2 cells, after we've fixed and stained them in 96 well plates. Now, these cells are particularly good for studying mitosis, as they only have a few and large chromosomes suitable for microscopic analysis. And they remain relatively flat. The cells remain relatively flat when they're grown as a cell monolayer, and the cells undergo mitosis every 2 or 3 hours, which becomes easy to capture the cells at various stages in mitosis. So we can see in the images represented on the right, cells undergoing prophase, metaphase, anaphase, cytokinesis, G2. Really impressive wide field, high resolution images using our 20, 40, and 60x long working distance objectives. Here's an example of a histology application where we're demonstrating color bright field imaging of chromogenically stained tissues where hematoxylin dye targets nucleic acids with that brilliant blue color, while eosin targets proteins with a pink color. Now, although entire slides have historically being saved for reprocessing, re-examination, and interpretation or analysis. With the advancements that we've seen in recent years in imaging technologies and data storage, colored digital imagery is really becoming much, much more desirable. And in this example or application, a whole slide with a series of prostate core samples was imaged at low resolution, and then augmented microscopy is being used to identify regions of interest, or let's call it ROIs, that were selected for imaging at higher resolution. And so multiple images were required to capture that, obviously, that entire region of interest ROI. And the montage of separate images were then stitched using Gen 5 software into a single high resolution image file, which is then used for downstream analysis. Here's another application where we're looking at changes in phenotype. So it's a phenotypic based workflow, assessing angiogenesis in transgenic KDRL M-cherry expressing zebrafish, which have been treated with various test molecules and VEGF inhibitors. And what we're doing is kinetically every 45 minutes over the course of 24 hours doing a Z-stack image with a 10x objective. And then with each image, we're placing an object mask around the entire vascular area and assessing the change in that area, change in phenotype, and assessing the angiogenic properties of the treatment that these embryonic zebrafish are undergoing. The last application I wanted to present to you, just to cover off the wide spectrum of capabilities of the Lionheart, was something in the microbiology space. Now, here we're looking at gram stain imaging. So gram stain imaging to classify, I would say, to classify bacterial species, both gram-positive and negative bacteria. So what we're doing here is, high magnification bright field and GFP fluorescence imaging using a 60x oil objective. Now, if you look carefully at the tile in the upper left corner of the panel on the right, this represents a digitally zoom image of a mixed bacterial cluster with both images overlaid. And the raw images were enhanced using digital phase contrast as well as background flattening and smoothing on the bright field image to improve the contrast between the gram-positive and gram-negative bacteria. And the two subpopulations were then identified and quantified using the architecture of Gen 5 software with a resultant specificity of greater than 99%. So as you've seen over the last 15 or 20 minutes, there's clearly a wide array and spectrum of applications and workflows that are possible using the optical architecture and software capabilities of Gen 5 and Lionheart system. And I would say my concluding remarks is that we have a technology or a platform here that provides diverse image acquisition capabilities, as I've alluded to over the last several slides. It's a modular and upgradable system that allows you to expand your footprint of applications even into the live cell imaging workspace. There's affordable options for those that don't need live cell imaging capabilities with the Lionheart LX system. A lot of sample flexibility and vessel handling flexibility with different types of vessel adapters. And the software itself, Gen 5, is comprehensive in nature, intuitive, of course, but comprehensive, that allows you to go from image acquisition all the way out to publication. And best of all, it's a compact benchtop system. Doesn't really take much of footprint space in the lab. Now, with that all in mind, what we really have is a system that allows you to study structure, function, and mechanism that hopefully leads to a broader array of discoveries. So with that said, I think it would be great to open out the call to questions, and we're here to answer your questions. So I'll hand the call back over to Bear.
All right, thank you so much for your terrific presentation, Charles. Now we're going to address some of the questions that came in. If you haven't already, please submit your question now using that Q&A module you'll find at the bottom left of your screen, and we'll get to as many questions as we have time for.
Wonderful, thank you very much, Bear. So I can see already some questions have come in, and maybe what I'll do is fire off the questions, and Maggie can take some of the answers. Right, so the first question I can see here is, so it says, I know black walled plates are best for fluorescent assays, but can I use the clear plastic plates we have in the lab with your instruments? So great question. Maggie, do you want to take that?
Yes, so it is a great question, and it's one that we get quite often actually in the field. Short answer, yes, absolutely. Our instruments are pretty plate agnostic. We'll take, pretty much anything that fits in like that standard microplate format. So I said short answer, yes. Black walled plates really are best for fluorescent assays because the black will absorb the light and can reduce background and, crosstalk between the wells, but if you're doing label-free proliferation assays, scratch assays, the clear plastic plates are perfect. And so, our recommendation is, don't go out and buy super fancy, expensive plates, try what you have in the lab and, adjust as necessary.
I think that's a great point, Maggie. And I will add to that to say, I think in almost all cases that works exceptionally well. Where I've seen glass bottom plates work exceptionally well is when you're going in the higher magnification. So once you're in the sort of 60 or 100x magnification, and trying to really get high resolution images. So some of the work we did with those PTK2 cells, looking at mitosis, that's where it would lend itself well to potentially using glass bottom plates, but it's not mandatory. We've had tremendous success just using polystyrene plates, clear or black walled clear bottom, either or. So that's the only other comment I will add to that. So the next question I see for you, it says, how does the Lionheart FX system perform when conducting kinetic fluorescence imaging on fixed XY coordinates? I want to image the same field of view over the course of the kinetic workflow. That's another great question, yeah, because kinetics is something we do quite a lot on the Lionheart, especially the Lionheart FX. So Maggie, do you want to take that?
Yeah, and definitely we understand the need to, follow the same field of view over the course of your experiment. Yeah, so when you're setting up an experiment in using our software Gen 5, you can pick where in the well you want to image. You can pick the X, Y, and Z, and those coordinates are saved within the software. And so it does return to those each and every time, different time points in your kinetic. And then the resolution of the stage of the Lionheart is really good. And so, you're not going to be, missing that cell or that area that's in the middle during the kinetic. If there is any slight movement, and I'm talking really, really slight movement, we do have a a processing step in our software that can align the images to make for a smooth movie for you when you're exporting your data.
Yeah, that's also, yeah, good point. And I will also add another comment to that. So the way that the Lionheart system is designed, it's a lead screw driven system. So especially when you're trying to do kinetic imaging, going back to the same XY coordinates, the resolution of where the image is captured from is incredibly tight. So it works really, really well, especially when you're trying to do time-lapse imaging of processes, cellular processes, or changes that are occurring over the course of, you know, whether it's minutes, hours, or days. I think that sort of hardware or optical design really helps achieve that. So yeah, great question and good points, Maggie. Thank you. So let's see, what else have we got here? Right, can users change the LED filter cubes themselves, or do we need someone from Agilent BioTek to come and help us? Great question from a user perspective. Do you want to take that?
Yes, yeah. Again, short answer, yes. Well, no, I guess the answer is no. You don't have to have someone come out and help you. So yeah, there are 4, positions for the LED filter cubes in our instruments, and they are 100% meant to be user changeable. It's part of so when your instrument is installed, part of that training is to make sure that you know how to not only use the system, but change those filter cubes, change out the objectives. It's actually just 4 screws, so it's really, really easy. There's 4, like I said, 4 positions, but a lot of labs have 5, 6 colors. And so it's really easy. You tell the software how you're changing it. And then again, it's 4 screws. It's really, really easy to switch it out and hardly takes any time at all. But it's something that users are meant to do themselves, but someone from BioTek will show you how to do it, so you're not on your own. We will show you first, but then absolutely something you can do, no problem, probably in your sleep after a couple of times.
Exactly. And on that point, I think I mentioned this earlier on during my presentation, there are over 24 filter cube combinations. So depending on what that target is, biological or cellular target you're trying to label, you tap into using the correct filter cube that's optimized with the ideal LED excitation light source, excitation filter, emission filter, dichroic mirror, so that you get good separation of light and exciting in the right spectrum of light and collecting light in the right area of the spectrum. So you've got over 24 different filter cube combinations to tap into and utilize, and then install the ones that are relevant. And as you have a busy lab where there's all sorts of different experiments going on, swapping out those filter cubes, as Maggie said, is easy as pie. It's really, really straightforward and easily done by yourself. So that's the only other thing I wanted to mention to you. So let's see, what else have we got? Another great question. So if we were to purchase a Lionheart and then had questions about setting up an assay afterwards, would someone at Agilent BioTek be able to help us? I think we can figure out how to set up imaging, but I'm concerned about the analysis. Great, because obviously quantitative microscopy is at the core of what we're doing with the Lionheart system, and being able to extract quantitative metrics from the images. So Maggie, do you want to talk a little bit about that question? I think it's a loaded question, but great thing to talk about.
No, it's great. We have a really, really great and strong team of field application scientists who do exactly what you're asking. Their role is to, not only initially train you on how to use it, but they are your go-to for, that first year when you're starting to learn the system for all of your scientific support. And so, in the days of COVID and everything, remote sessions seem to work really well when we can just log into your, remotely access your computer with you, and like walk you through how to help you set up your analysis, or help troubleshoot things that you've already done. In-person trainings can happen as well, but short answer is you'll have a dedicated FAS, field application scientist, that would be working with you, and you can email with questions. We also have a very good technical support team for questions that are beyond analysis, but maybe it's I don't remember which button I'm supposed to press to do something. A bunch of people in Vermont can help you answer those questions as well. So there's a lot of people that you can ask questions to, and our goal really is to make sure you're fully supported, make sure you know how to use your instrument, to the fullest capacity. We want it to be used and used correctly, and so it's just an email away usually, and you'll have your help.
Yeah, and all very valid points. And I actually do once again want to extend a personal experience that I've had working with one of many customers around the world. Is we've worked on an application, so the DNA damage repair application, we talked about the COMET assay. So that was done in partnership or in conjunction with one of our customers who wanted to develop that application pretty much from ground on up. And, members of our FAS team and organization and applications lab were intimately involved in dialoguing with that customer, even in many cases coming to the lab and helping them optimize parameters of sample preparation and the entire workflow, even before it's even got the system for imaging. So we really do want to be actively involved, helping you to optimize and streamline the process and the workflow and get you all the way out to publication. I think it's in our mutual interest to do that, and we love providing scientific and intellectual input and helping folks get to where they need to get to as easily as possible. So that's another sort of word of that I wanted to sort of share with you. There is another question that's come in. It's a simple question. It just said, what is the learning curve? I think it's sort of tied to the previous question indirectly, but again, having had personal experience come from the arena of quantitative microscopy and high content screening, most people may think, well, the learning curve is pretty steep, but Maggie, do you want to take a few words on that? I think that would be something very, very important to address.
Yeah. So it's hard. It's a hard question to answer too, because it really depends on your level of experience. People that do have experience with automated imagers or microscopy in general, the software it's different than other softwares out there, but it's the same in so many ways, and so you would be able to figure it out on your own. That being said, I have a number of customers that are undergrads that are using it in their lab, and so you don't need to have a master's or a PhD to work our software. Undergraduates can use it. We generally, if I'm going in there and training a customer, it's like a half a day of training to get them up to speed, and then there's normally like a follow-up training later, and then people are usually they're good to go. They interact with us not nearly as long as you would think. So I'd say the learning curve is not that steep comparatively. The software, will walk you through what you need to do next, and We have great training manuals. We have training videos online that you can watch for different things. You have your FAS support and your technical support. And so if you're struggling more, you have a good backup system as well to help you out.
I'm going to add a little bit of subjectivity to it. I apologize for that. But obviously, having been an end user and also a member of our support organization, there's two phrases that come to mind. And again, this is my subjectivity coming into play. One is intuitive, and the other one is short learning curve. That's my interpretations, but it's also listening to what a lot of the users have been saying, especially, let's say, grad students who are new to using our devices or new to our user interface. That's the sort of general feedback I'm getting. So I think the best way to put that to the test, as I say, the proof is in the pudding. So is to, sort of reach out to us and we can do, a software demo with you, or bring in a Lionheart system so you can actually see it in operation in the context of your own applications and your own workflows. So that's the only other comments I wanted to add. Yeah, so I'll leave it at that. I think we need to wrap up because we're coming to the end of our timeline. I just want to say thank you to everyone for tuning in and listening to our talk, and sort of asking Maggie some wonderful questions. And we really do look forward to hearing from you. Hopefully the content of our Q&A and the slides was thought-provoking, got your intellectual juices flowing, and you guys want to now maybe think about how you could utilize these sorts of workflows and technologies in the context of your own research initiatives. So I'll leave it at that. Maggie, any closing comments before we hand it over to Bear?
No, I think you covered it perfectly, Charles. You have the energy to do it. So.
Thank you very much. Well, we wouldn't have done it without you, so thank you immensely.
Welcome.
Great. Well, thank you both. I just want to thank Dr. Pruitt and Dr. Amir Mansour for a great presentation and discussion. And on behalf of Agilent Technologies, I just want to thank every one of you that joined us today. Please know that a recording of this webinar will be emailed to you in the coming days. And if you would like more information on Agilent BioTek's products and services, including our archive of previous webinars, please visit us at www.agilent.com. We hope that you enjoyed today's presentation and have a great rest of your day.